Hello students, welcome to BioSmart Academy. In today's lecture, we will continue our lecture series on developmental biology module, which is a very important module for CSIR NET exam, and we will provide free lectures for your CSIR NET June 2021. We try to cover all the modules with important topics. Okay. So we already uploaded two videos on developmental biology. One is on apoptosis in C. elegans and another one is on valval formation. Okay, in C. elegans. So we already upload these two videos. So if you not watch these two videos till now, then please go and watch these two. Now today we will going to start a very important video of developmental biology. Okay, and this is the axis formation in Drosophila. Okay, and remember student, this is the part 1. This is a part 1 of this video lecture, axis formation in Drosophila. Okay, remember one thing is that the axis formation in Drosophila, first of all, very important topic from examination point of view. Second point is that this is a, you can say, one of the biggest topic in developmental biology, from where you definitely get 2 to 3 question, okay, every year on CSI net, that is of 4 marks. Okay, this year also asked 2 to 3 question. Okay, and the third important thing is that we will try to cover this whole lecture of axis formation in Drosophila in several parts. Okay, so this will be the part 1 of this lecture. So I just recommend you that please follow our lectures, you will definitely get benefited. So this will be the part 1 of this lecture, and we will try to complete this whole lecture in several parts. Okay, so please follow us, please share our BioSmart Academy YouTube channel, which is a very important, you can say, YouTube channel will provide you free lectures on YouTube for CSIR NET, GATE, DBT and ICMR. Okay, so please share and subscribe. Now let's start today's lecture, that is part 1 of axis formation in Drosophila. Okay, so first of all, in this lecture we first discuss about that how the basically that why Drosophila is chosen, or why basically what are the basically changes basically is came as basically Drosophila basically a difficult organism on which we can study embryology. Okay, so we will discuss basic some things in this part. Okay, so basically, first of all, as you can see, this is the picture of Drosophila. Okay, this is a picture of Drosophila. So Drosophila, why Drosophila is chosen? Basically, there are mainly 4 points. Okay, please note down the things that I discuss here. Okay, because Drosophila is easy to breed. This is the most important thing. Easy to breed. It is basically hardy because of its structure. It is basically prolific. And the last point, that is the most important, that it is tolerant of diverse condition. It is tolerant to diverse condition. Okay, so in any condition you can culture it, or basically you can say that it is basically tolerant to diverse condition. So these are the major 4 factors by which we can use Drosophila as our genetic basically model. Okay, where we can study its genetic, its basically embryological parts. Okay, so these are the 4 points. Okay, now another important point is that regarding Drosophila is that in some larval cells of Drosophila, DNA replicates several times. Okay, DNA replicate, that is a DNA basically replicate several times. DNA replicates several times without separating. Okay, so do you know what this phenomena is known as? This phenomena is known as polytene chromosome. Okay, we basically known this phenomena as polytene chromosome, where basically hundreds of strands of DNA are adjacent to each other. Okay, polytene chromosome. Okay, many students who basically of zoology or any other basically field, you can say biotechnology, they basically done practical on this polytene chromosome. Okay, they basically done practical on this polytene chromosome. Basically they are basically used to take the larval stage of Drosophila, and they just cut out their basically the mouth portion, and from there you basically stain the chromosome, and you were basically seeing this type of structure. This is a polytene structure. Okay, this is the polytene structure of chromosome. Okay, where you can see that basically this basically is taken from the salivary gland cell. Okay, basically salivary gland cell. Okay, you know very well, we basically separate this salivary gland cell where we see this polytene chromosome. Okay, and each polytene chromosome has 1024, that is 1024 strands of DNA. Okay, and this DNA are basically stained here in blue color. These are the DNA. Okay, like this, these are the DNA strands. These are the DNA strands. Okay, that is strands in blue color, you can see here. Okay, these are the DNA strands. Yeah, all this. Okay, so in polytene chromosome, each polytene chromosome, you can told out that each polytene chromosome contain or has 1024 strands of DNA. 24 strands of DNA. Okay, so this is about the polytene chromosome. Okay, so these are the basically DNA strands. This blue in color, this blue is here, all this. So this is the structure of the polytene chromosome, and this is the structure of Drosophila. So this is a basic introduction about the Drosophila. Okay, now we shift our basically discussion to the its embryological part. Okay, we shift our discussion to its embryological part. Okay, now remember one point: to study the embryology of Drosophila, it is very difficult. Okay, why it is difficult to study the embryology of Drosophila? Because of its complex and complex structure. Because if we try to experiment it, okay, or if we try to see observe it microscopically, it is quite difficult. Okay, and it makes our basically easier to study its embryological part with the help of molecular biology techniques. Molecular biology techniques. Okay, molecular biology techniques. Clear? Okay, molecular biology technique, or you can say through the manipulation of the insect's gene and RNA, or you can say the manipulation of insect's gene and RNA. Okay, RNA, that its genetic could be related to its development. Okay, so this revolution basically is basically a large part because of the this revolution is continuing, and in large part because why? Because of the availability of the complete due to ability of the complete genome of Drosophila. Okay, simply, so as we have the complete genome of Drosophila, so we can easily manipulate its gene, its RNA, and with the help of this molecular biology technique, we can easily study its embryological features. Okay, clear? So we can easily study its embryological features. Now, so the things that I discuss here, okay, this all are the basically the basic part of the Drosophila, or you can see the introduction, or you can call it at the introductory part of the Drosophila. Okay, now the major part that now we're going to study about the genetic mechanism, or you can say the patterning of the Drosophila body. Okay, that how the Drosophila body is formed. Okay, so basically here in this we will going to study the segmentation and the anterior posterior body plan. Okay, so So see, one thing is that we all know the process of embryogenesis begin at fertilization. Okay, the process of embryogenesis, the process of embryogenesis began at fertilization. Okay, the process of embryogenesis began at fertilization. Now, but many of the molecular event that is critical for Drosophila embryogenesis. Okay, but there are many events which are basically critical for Drosophila embryogenesis actually occur during oogenesis. Okay, during oogenesis. Clear? Now, so before the oogenesis began, sorry, before the oogenesis began, the oogonium. Okay, before the oogenesis began, the oogonium. So what is oogonium? Oogonium is basically a oocyte. Is basically formed from a single female germ cell. Oogonium is basically formed from a single female germ cell. So before the process of oogenesis began, the oogonium is basically what? This oogonium is divided 4 times with incomplete cytokinesis. Divide 4 times with incomplete cytokinesis. Incomplete cytokinesis. Okay, with incomplete cytokinesis. Clear? Now, and it gives rise to 16 interconnected cell. Note it down. It gives rise to 16 interconnected cell. 16 interconnected cell. Okay, now this 16 germline cell along with this, okay, now this 16 germline cell, okay, there are 16 germline cell, and along with the surrounding epithelial layer of somatic, okay, these 16 are what? These 16 are germline cells. These 16 are germline cells, and along with the somatic follicle cell, that is surrounding the somatic follicle cell. This is somatic follicle cell in yellow, constitute the egg chamber in which the oocyte develop. Okay, so this both constitute the what? Egg chamber. Okay, egg chamber. So basically this is 16 germline cell and this is basically what? Somatic follicle cell. Okay, and both of these contain what? Both of these contain egg chamber. Clear? And in this egg chamber, oocyte will develop. What happened? Oocyte is develop. I hope student you get till now the clear idea. Okay, oocyte is developed. Now, okay, now this germline cell includes 15 metabolically active nerve cell. Okay, now we're going to study about the germline cell. Now these germline cells, okay, if we consider this germline cell, okay, these germline cells, okay, include what? 15 nerve cell. This germline cell include 15 nerve cells. Okay, basically metabolically active nerve cell that make mRNA and protein. Okay, what does this nerve cell do? The nerve cell make mRNA and protein. Clear? Note it down. Okay, so mRNA and protein that are transported into the single cell. Okay, that are transported into the single cell that will become oocyte, and this mRNA and protein basically transported to a single cell that become oocyte. Clear? Okay, now as the oocyte develop, okay, at the posterior end, okay, so where this oocyte develop? So this oocyte develop at the posterior end. As the oocyte develop at the posterior end, okay, of the egg chamber, So numerous mRNA made in the nut cell are transported along microtubule. Okay, so this numerous mRNA that basically made in the nut cell are transported along the microtubule, through the cellular. Basically there are some interconnection there into the oocyte. Okay, so basically as the oocyte basically develop into the posterior end, so the nut cell that basically going to form mRNA, that mRNA going to transport it to the oocyte by the process of microtubule. Okay, we all know that kinesin and dynein, they basically transported to the oocyte. Okay, so basically this is basically a basic introduction about the how the basically nut cell form basically this mRNA and protein, and how these mRNA and proteins are basically transported to the posterior end through the microtubule to the oocyte. Okay, now so now till now I hope that you got a clear idea. Okay, now so basically here we were going to identify the genes that is first established anterior posterior polarity, established anterior posterior polarity, and second divide the embryo into specific number of segment. Okay, divide the embryo into a specific number of segment. Okay, with different identity. Okay, divide the embryo into specific number of segment, into specific number of segment. Okay, with different identity. Clear? So basically we have to basically identify a hierarchy of genes that basically establish the anterior posterior polarity, and secondly divide the embryo into specific number of segment and each with different identity. So the hierarchy basically what? This hierarchy basically initiated by maternal effect genes. The hierarchy basically initiated by maternal effect genes. So the hierarchy basically initiated by maternal effect genes, and this maternal effect gene going to produce mRNA. The maternal effect gene going to produce what? mRNA that is localized to different region of egg. Suppose this is a egg, a egg, and this mRNA is basically located to different regions of the egg, okay, different regions of the egg, because it is a maternal effect genes. And you know in maternal effect genes basically it is basically cytoplasmically distributed the whole mRNA. Okay, now this mRNA, okay, now just write it down, this mRNA. Okay, so here mRNA is produced. Okay, clear? Now this mRNA is basically encode some transcriptional and translational regulatory protein that diffuse through the blastodome. Okay, encode some transcriptional and translational okay, encode some transcription and translational regulatory protein, some proteins. Okay, regulatory proteins. That regulatory protein means what? It means they can regulate, either activate or basically repress the expression of some zygotic genes. So basically maternal effect genes produce mRNA, and mRNA basically going to basically produce, or you can say mRNA going to encode some transcriptional and translational regulatory protein, and this regulatory protein going to activate or going to repress the zygotic genes. Clear? Now, so what are these zygotic genes? So the first zygotic genes that is expressed is GAP gene. Just note it down, GAP gene. Then I will show you the figure of GAP genes. Okay, so this is the first zygotic genes. Okay, why it is known as GAP gene? GAP gene because mutation in them cause gap in the segmentation pattern. Because mutation in them cause gap, cause gap in the segmentation pattern. That's why it is known as gap gene, because of the mutation in them cause basically gaps in the segmentation pattern. Okay, so this is a figure of the gap gene. Here you can see the figure. So here in this figure you can see this is a cytoplasmic polarity, it is the maternal effect genes. So this is a maternal effect genes, or you can see cytoplasmic polarity, where you can see basically mRNA, basically they produce and mRNA are basically localized, and here this mRNA going to basically encode some regulatory protein, as I discussed. Okay, and this regulatory protein basically going to activate or repress some zygotic genes, and these all are basically zygotic genes. Okay, that is the first we discuss gap gene. So this is a figure of gap gene. Here you can see this is a gap gene, and in this figure you can see that there are some gap. This is a gap, this is a gap, this is a gap, and this gap is occur due to the mutation in the segmentation pattern, that is known as gap gene. Okay, now this gap gene are expressed in certain broad partially overlapping domain, and then gap gene encodes some transcription factor. Now this is a gene, gap gene. Then what happened, it encodes some transcription factor, that is TF. Okay, transcription factors, and by different combination and concentration of gap gene, it regulates the transcription of pair rule gene. So this is the pair rule gene. Okay, so basically gap gene produce some transcription factors, that is TF, and it basically control the expression of the pair rule gene. So this is the pair rule gene. Okay, clear. Now pair rule gene is basically formed. Okay, and in pair rule gene you can see it divide the embryo into periodic units. So this is 1, this is 2, this is 3, 4, 5, 6, 7. So in this it basically divide into 7 basically seven band, that is perpendicular to anterior posterior axis. You can see here, clear, perpendicular to anterior posterior axis. Now the transcription factor. Now so pair rule gene, so now what happened, so pair rule gene basically encode another transcription factors, that is TFs, similar that of gap gene. Pair rule genes also encode some transcription factor, and the transcription factor encoded by the pair rule gene activates segment polarity gene. So where is segment polarity gene? So this is the segment polarity gene. Okay, clear. So this is the segment polarity gene. Okay, and here you can see that in segment polarity gene, basically the embryo is divided into 14 segment. So if you count this 1, 2, 3, so like this it basically divided into 14 segment. So in segment polarity gene, the embryo is divided into 14 segment, and it establishes the periodicity of the embryo. Okay, so at the same time, if you look at this figure once again, okay, if you look at this figure once again, at the same time the protein product of gap gene, okay, just note it down, the protein product of gap gene, okay, pair rule gene, okay, segment polarity gene, okay, interact, they all three interact. Okay, so these all three interact, to regulate another class of gene, and and this class of gene is known as homeotic genes. So this class of gene is known as homeotic gene. So here you can see, so this is a figure of this is homeotic gene. So this is a figure of homeotic genes. This is homeotic genes. So what are the homeotic genes? The homeotic genes basically determine the developmental fate of each segment. Okay, so homeotic genes basically determine developmental fate. Okay, developmental fate. Okay, just I write down here, just note it down, that is the gap gene plus pair rule gene plus segment polarity gene. Segment polarity gene. So all these three control the regulation of another class of gene, that is known as homeotic selector. genes. Clear? And this homeotic selector genes, what does it do? It basically determine the developmental fate, the developmental fate of each segment. Okay, so it determines the developmental fate of each segment. So now note it down and just look at another time for the figure. Okay, so basically, so from this figure, so this is the homeotic gene. It basically determine the fate of each segment, and these three gene, gap gene, pair rule gene, and segment polarity gene, control the expression of homeotic gene. So students, that's all for today's lecture. This is the part 1 of the axis formation in Drosophila. So here we discuss some of the zygotic genes and some of the basic introduction of Drosophila. Okay, in our next lecture, that is in the part 2 of the video, we will going to discuss in brief about the anterior posterior polarity in oocyte. So please share our YouTube channel. We will going to cover all the important topics, all the important module in free. You will get on YouTube and crack the CSI June 2021 with us. Thank you very much.